All right, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 91st seminar, and I call it a Caltech Day. It's been almost 18 years since I came to the United States for the first time to study at MIT as a PhD student. It has been an interesting and tough journey as a foreign person with a lot of ups and downs. Although international students and scholars' treatment and status have been improved over the years, there are many things to improve further to increase the benefit of diversity and ensure human rights. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Mikhail Shapiro. I mean, he doesn't need any introduction because we all know him. He is a Caltech professor who has published numerous high-impact papers, including one of my favorite papers, especially on imaging of living systems. That's amazing. If you didn't read his paper, you should read many of them. This year, he served as one of the conference chairs for SEED, the premier synthetic biology conference that my advisor, Chris Boyd, started in 2014. We also had a great dinner discussing ASA synthetic biology's future with other editorial members and editor-in-chief, Humin Zhao. And he also suggests many interesting new things. And then I respect, his vision, his kind of idea, and everything. Mikhail, thanks so much for your innovative research and enthusiastic education effort for the future leaders, and the virtual podium is all yours now. And thank you so much again.
Thank you. Thank you, Taesuk. Really a huge pleasure to be here. I'm honored to be invited as a pioneer and to be the warm-up act for Alex. I think this is such a great idea, what you've organized with this series, and really look forward to hearing today's talk. So I'm going to just give a couple comments. I was asked to talk about kind of big, inspiring vision, and really, I just want to show 3 slides to just credit my lab. So maybe if I may just briefly share here. This is just a picture of our group from last year's beach party. And I've just been very lucky at Caltech to work with such a tremendous group of people. I think I have PhD students from 6 different graduate programs currently. And I think the cool thing about the synthetic biology field is that depending on what we're working on, it really requires participation from people from all kinds of different disciplines, and is like a central meeting point for these disciplines. So I really appreciate that about synthetic biology. And since I have 3 minutes, I wanted to give a plug for one particular technology that we developed in our lab that's kind of was developed based on need that we had, but I think might be useful to the broader community. And that's this thing we call SEMPER that was developed by Tom Dwan and Ishan Dev and colleagues in my lab. These are 2 graduate students, one in bioengineering, one in chemical engineering at Caltech. And the idea is very simple, and it came out of our need to create tunable polycistronic expression in mammalian cells. So let's say you want to express 2, 3, or more genes together. Currently, you have either 2A peptides or IRESs that most of us rely on. 2A peptides nominally have a 1-to-1 stoichiometry, but it can vary a little bit and is maybe difficult to control. IRESs give you control, but require some space. And so we thought, there might be something simpler. And so what Tom and Ishan came up with is something very simple. It's basically saying, I'm going to take genes and just focus on these 2 GFP and BFP genes here, and just put them back to each other under the same promoter. And then what I'll do is I'll have a not fully strong translation initiation sequence in front of the first gene, so that some of the ribosomes will skip through and read the second one instead. Okay, and by tuning the strength of this translation initiation sequences, you can create tunable expression levels for these genes, and it works even better than we expected. I should credit Clifford Wong, who originally developed these leaky TIS-based siphoning system, these so-called upstream ORFs, which were just very little ORFs that were used to tune the expression of a downstream gene. But the idea here is you can put an actual ORF that you want the product made of in the front. And this actually works not just for tuning 2 things, but 3 things, and potentially could be expanded even more. So I just thought this might be something useful to the community. It has some caveats, like you have to remove methionines currently from these genes, but we found that we've been able to do that, and this has been very helpful for us in expressing our gas vesicles and maybe useful to others as well. And here's a link to this at the bottom. So that's my 1-minute advertisement. I won't take any more time away from what I'm really looking forward to with Alex's talk.
Well, that is wonderful. So you are now also, I mean, you are doing everything. I mean, in many sense, like other big names. I mean, that's fascinating. So you are kind of, your direction is kind of moving toward some other area, not only imaging in that case.
Well, it's kind of like out of need, right? I don't want to invent new synthetic biology tools, but I have to. Or we have to in our lab, because what we want is not available. And I think that's what's really cool about our field, is that it actually pushes us to develop new stuff across different areas, because we need it to accomplish our synthetic biology goals.
That's wonderful. That remind me of, I'm also engineer. Also, I work because I need something, and then I remember Howard Salis developed RBS calculator because we cannot predict the strengths of the RBS, and then we cannot tune the expression level. And then that is actually side project and become the main one. And then that makes Howard and Chris Boyd kind of famous at the end of the day. That's amazing. Thank you so much. I mean, I really looking forward to really seeing your paper published in the big journal later on.
Thank you.
Okay, thank you. All right, so okay, so now the main speaker of today with our longer introduction, Alex Vlahos, is currently a Human Frontier Science Program Fellow in the laboratory of Dr. Xiaojing Gao at Stanford University. So that means both speaker from the California. He applies principles in synthetic biology and protein engineering to develop tools for programming intercellular signaling. Previously, he completed his PhD in biomedical engineering at the University of Toronto, where he used principles in tissue engineering, regenerative medicine, and biomaterials to develop platforms to improve vascularization of the subcutaneous space for inlet transplantation. His goal is to convert his background in synthetic biology, systems biology, and tissue engineering to mechanistically study multicellular systems such as the immune system. His future lab will lay the groundwork for designing smart therapeutics using synthetic protein circuits to engineer programmable cells that dynamically respond to changes in the local microenvironment, model complex intracellular interactions, and then engineer specific therapeutic responses for biomedical applications such as improving cell therapies. Alex, thank you so much for your time today, and the podium is all yours, and please take it away. Thank you.
Okay, so thank you so much for the introduction, and also Mikhail, I think you framed sort of the importance of synthetic biology in such a great way, and I came to the field of synthetic biology with the need to sort of create these tools. So my background is in tissue engineering, and what I'm showing here is an example of a transplanted islet. And so islets are the cells that regulate your blood glucose. And in individuals with type 1 diabetes, the insulin producing beta cells are degraded. So, they can regulate their blood glucose using exogenous insulin, but it really can never fully recapitulate what a transplanted cell can do. And, the field of tissue engineering has really focused on 3 principles or approaches to sort of be able to develop functional tissues to support, to regenerate either an organ or support some sort of biomedical need. In engineering materials, specifically the scaffolds, engineering the cells, and then engineering specific tissue architectures. And really, this is all with the goal of mimicking physiological properties of tissue. But fundamentally, whenever you transplant anything, there are 2 barriers that need to be overcome. One is sufficient vasculature to support the transplanted cells. And the second issue, which is probably the more pressing one, is protection from the host's immune response. And so, people have really tried to shift away from doing systemic immunosuppression because, one, for individuals with type 1 diabetes, often it's referred to as juvenile diabetes. So having a child have an islet transplantation and then have to be on systemic immunosuppression for all these years is not feasible. And there's a lot of secondary complications that can occur. So there's been a huge push in the field of tissue engineering and regenerative medicine to sort of look at local immune modulation approaches. And one of the when I was looking at trying to figure out how I'd want to, where I'd want to go for my postdoc or do for my postdoc, the 2 big approaches that the field of tissue engineering use for immune modulation is you can either genetically engineer a cell to overexpress a protein of interest or knock down a specific signal, or you can do more of the biomaterial based approaches where it's like the traditional drug release profiles. And they both have their advantages and their uses, but fundamentally, when you think of the immune system is a very dynamic beast. There's a lot of different cells in play. There's currently no silver bullet to completely turn off the immune system. So, that's why I kind of err away from just simply overexpressing. And although you can have more programmable capabilities using these biomaterial-based approach, supramolecular or whatnot, a lot of these approaches have a finite reservoir of immune modulating biologics. So I came to synthetic biology with the goal of, well, what if we can take the best of both worlds, where we have a cell that in theory can produce a limitless supply of immune modulating growth factors, but we also want to have the capabilities and programmability that you might have with maybe more of the biomaterial based approach. And so that's what really gravitated me towards synthetic biology, where the idea is that you can take an engineered cell, have it sense, interface, and respond to complex biological systems such as the immune system, with the goals of biasing specific host responses and improving cell transplantation for the long term. And so when I looked at the fields, traditionally the field of synthetic biology has really focused on these traditional gene circuits, which are great. They have a lot of power. They have the ability to interface in many different systems. But one thing that kind of always got me was the fact that they could be, sometimes implementing them can be a little bit cumbersome, in the sense that you have to integrate a lot of different components into the host genome in order for them to function. So in the past 5-6 years, there's been a new field that's emerged, which is these synthetic protein circuits. In part, my postdoc supervisor Xiaojian Gao was one of the first to show sort of the CHOMP system, which I'll talk about. And I'm not trying to say gene circuits are bad. Just in different situations, you might have different advantages and disadvantages. And one thing that really interested me with the synthetic protein circuits is fundamentally that it can directly interface with the normal signal transduction pathways of a cell, which function at the protein level. And secondly, and something I really have focused on, is the fact that these protein circuits in theory can be quite compact, meaning that you can encode them all within a single transcript, and they have the potential to be delivered via mRNA depending on what your application is. Something in which DNA circuits require a direct integration. And so what, I really got enamored with the field of synthetic biology when I first actually saw Xiaozheng give his job talk at the University of Toronto. And he talked about, the fact of using a platform called CHOMP, which stands for Circuits of Hacked Orthogonal Modular Proteases, that he developed during his time in Caltech with Mike Elowitz. And really, it was a really nice, elegant design where you simply use orthogonal proteases which cut at specific sequences to communicate with each other. And he showed that you could generate these regulatory cascades, binary logic gates from the point of view of decision making, and finally analog signal processing. And this was something so cool to me when I first came and saw. And so when I looked at the field and thought about transitioning to SynBio, I realized that proteases actually have become a common currency for many protein circuits. If we think about a lot of the different synthetic receptors currently out there, a lot of them use protease activity, to release a transcription factor and activate a specific gene of interest, but fundamentally they still rely on protease activity. So you can imagine these as the inputs, right? Where if you took a protease, split it, it'd be inactive, and then in response to some sort of ligand, had a reconstitution of the activity. And now we had, this processing module in CHOMP. But the one thing that was, lacking that I realized is the fact that all of these proteases that CHOMP functions in the cytosol, and you can program and engineer a cell of interest, with this processing capabilities, but you had no way to actually communicate with other cells, simply because when you think of the biology of a cell, you have translation of cytosolic proteins is very different than translation of secreted proteins, right? And so I realized there's a need to engineer a general approach to control protein secretion at the post-translational level if we had any hopes of trying to create these engineered cells to communicate with the local microenvironment. And so I kind of alluded the fact that, when you go back to the biology, normally cytosolic proteins are translated directly into the cytosol. They lack a signal peptide, which is found in pretty much all secreted proteins. And this signal peptide is recognized early on in translation. And what happens is for any secreted protein or membrane protein, typically what happens is it gets co-translated directly into the ER. And this ER is, you can think of it as a separate barrier. So you can do all the engineering you want in the cytosol, but how do you actually communicate with something that's normally secreted? And funny enough, when I was writing my thesis, I realized that, the beta cell, which I've been, studying and working on transplanting, actually does this sort of post-translational control of protein secretion. And I'm not going to go into the details of the mechanism, but essentially what it does is, it can pre-package proteins into vesicles, and then in response to a given stimulus, it promotes their secretion. And so this is kind of a unique property of neuroendocrine cells. You can imagine neurons do the same thing with neurotransmitters. But what we wanted to do is, create a general approach that works in every cell. And, I always, anytime I look to design something, I always like to make sure that I've thought of what are the main criteria that I want to do, because, I find if you just try to design on the fly, it's not ideal. So, the first criteria, and probably one of the most important, is that I didn't really want to re-engineer CHOMP. We know CHOMP works. So I needed a strategy that, I can control the secretion through the cytosol, which is probably the most important thing. I wanted orthogonal control of secreted proteins of interest, meaning that only when I activated the circuit would I get protein secretion. I needed to make sure that I didn't, mess up the natural protein secretion machinery of the cell. And finally is I wanted this platform to be generalizable, right? One of the biggest issues I find is that, if you design something that it only works in a very specific case, that really limits the utility of the tool. And so, we came back to the conventional secretory pathway, and we realized that whenever proteins are translated and secreted proteins are translated, as they normally go through the secretory pathway, they're going to be just constitutively secreted. They'll go from the endoplasmic reticulum to the cis Golgi, eventually to the trans Golgi, and then into the plasma membrane and secreted. And this is really driven by this anterograde transport. But we realized that there are some proteins that are ER resident proteins that have these ER dilysine retention motifs that are recognized in the early Golgi apparatus, and they're transported back into the ER and effectively retained in the ER by these specific motifs. And so one key thing, though, is that these motifs only work for membrane proteins, but something that we realize that could be to our advantage is that these motifs are cytosolic facing, meaning that they're not sequestered from the cytosol and they're directly exposed to the cytosol. So, this strategy might work for membrane proteins, but what about secreted proteins? Funny enough, we also realized that in the trans Golgi, there's actually a native endoprotease called furin. And furin is a protease that is often involved in converting pre-proteins into their mature functional state. And one really nice sort of characteristic of furin is that it's only expressed and active in the trans Golgi apparatus and in the plasma membrane, so far down the conventional secretory pathway. And so the nice thing too is that it cleaves at very specific recognition sequences. So, we realize that there's two components of the conventional secretory pathway that we might be able to engineer here. And so our first goal was to show, what if we took a normally secreted protein, in this case, SEEP, it's just simply a reporter protein. And if we just made it membrane bound, put a furin cut site in the interior linker and this retention motif on the cytosolic phase, can we actually, prevent any sort of secretion from occurring? And so what we saw is that, when you have the retention motif present, the amount of SEEP secreted into the supernatant was quite low. And when you modify it and get rid of those two key residues involved in retention, you end up getting really robust secretion of the protein of interest. And so, it's kind of obvious where the next step is, where instead of, worrying about or, doing some sort of mutagenesis, which is not feasible from a activation standpoint, what if we simply used proteases to sort of remove this retention motif whenever we want it? And so that sort of spurned the platform called RELEASE, which stands for Retained Endoplasmic Cleavable Secretion. And really the way it works is that we just put simply a protease cut site between the transmembrane domain of the protein and the retention motif. And what happens is that you can tag any protein of interest that normally would be secreted. You can imagine a cytokine, a growth factor, or even membrane-bound proteins by simply removing the furin cut site. And this protein of interest would be normally retained in the ER until you have activation of your circuit, either a protease sensor or the CHOMP circuitry, where the retention motif would be removed. And then this protein of interest would then be transported through the conventional secretory pathway. And then once it reaches the trans Golgi apparatus, that's when it'll be cleaved by furin into its secretable form. So, this secretion is really dependent on that initial protease event. And what we just simply showed first is that, when we co-expressed the protein of interest with and without our release construct, we can get really nice control over protein secretion. And the really nice thing about proteases is because they cut at very specific cut sites, you can imagine that by simply swapping out the protease cut site, you can now make orthogonal release constructs to control the secretion of different proteins of interest. So we created a TEV inducible and an HCVP inducible release setup. We also wanted to determine if we could implement processing capabilities directly at the release module. So you can imagine release in the protein circuit toolkit as the output module. But there might be some ways that we can implement some sort of logic gate. So in this case, when you have a pro-inflammatory macrophage that might be secreting a bunch of different pro-inflammatory signals, you might have a case where you want to implement some sort of logic so that, let's say, if either IL-1 beta or TNF alpha is present and you had a sensor for that specific thing, you would create essentially an OR logic gate. And this one is relatively simple to do, where you just put two proteases in tandem, cut sites in tandem, so that if either or of these sensors or proteases are activated, you would have removal of the retention motif. We also implemented AND logic, which is a little bit trickier to do, but we found that there were actually some N-terminal retention motifs as well that we could harness and use, so that in order for you to get secretion of the protein of interest, you would actually require both of these proteases to be activated or signals to be present, and to cut those respective retention motifs and control protein secretion of your protein of interest. And so all I've shown you now is sort of the idea of using reporter proteins, but what about functional biologically relevant proteins? And so one of the ones that we were really interested in initially was being able to control the surface expression of KIR 2.1, which is a potassium channel that is involved in many different neurobiological applications. And there's sort of two criteria that made KIR 2.1 an interesting design challenge. One, that it's functional as a tetramer. So, if typically the release platform would work on the monomer, right? And then the other thing was that KIR 2.1 has an ER export signal built in. So we wanted to determine if we could actually overpower that export signal with our retention motif. And so we also genetically encoded a voltage sensor. So when the membrane would be hyperpolarized by the expression of functional KIR 2.1, what would happen is that you'd have an associated colorimetric change, an increase in fluorescence intensity. So our first goal was to see, okay, can we get KIR 2.1 on the surface? The KIR 2.1 that we used had an HA antigen that we could stain. And we saw that, when you co-express the protease, you end up getting an increase in the surface expression. But more importantly, we found that the KIR 2.1 that we had on the surface was also functional. So it was really nice to know the fact that even though we're modifying this channel at the C terminus, it still remained functional and was able to sort of drive this membrane hyperpolarization of the cells that we transplanted. Another thing that we were really interested in is one advantage of these protein circuits is that you could theoretically develop sensors for different intracellular states. So one sort of protein that is quite, RAS is an oncogenic protein implicated in a lot of hard to treat cancers. And so during Xiaozhang's sort of postdoc, he developed a RAS sensor based on simply a binding of the RAS binding domain of CRAF, which is another protein downstream, that he had attached a split TEV protease to it. And the idea is that when RAS is active, you'll have recruitment of this RBD binding protein, reconstitution of the protease activity, and then activation of the subsequent circuit. And so our first design setup ended up really failing miserably. And we kind of realized that the reason is that this protein dimerization event is occurring at the membrane, but the ER platform or the release platforms at the ER. So we have sort of this spatial issue that how do we propagate a signal from the membrane to the ER? And so what we simply did is we realized, well, what if we just put a relay protease in between? I'm not going to really focus on the details of how we did it. It's in the paper, but we simply our goal was to propagate a signal from the membrane and drive, through protease activity, have it localized to the ER using sort of a split protease approach. And in this case, we showed that, you can start to begin to develop these novel input-output functions where in we have a RAS sensing circuit, and then what you're secreting is IL-12, which is a pro-inflammatory cytokine. So you can imagine, in this case, you could deliver this and the cancer cells would be expressing a pro-inflammatory cytokine to drive an inflammatory response. And so we've now, this is what I imagine the new synthetic protein circuit toolkit looks like, where you still have these sensors, the information processing both at chomp and release, and then fundamentally now you have some nice extracellular outputs. But this was the first generation release, and that's great. I'm always looking at ways to improve it, simplify it, and really look to translate it. And so this idea of polycistronic designs is really quite an advantage of protein circuits over these genetic circuits, but the current setup of release means that it only would function as the final protein in the polycistronic case, because the retention motifs we're using must be at the C terminal, right? So this severely limits your capabilities, because what happens if you want to use multiple release constructs, right? What happens if, you know, you want to put a reporter protein, or you know, it really complicated the whole system. What we realized, though, is that there are actually other retention motifs out there, and one that really sort of interested us was this RXR motif. And the one really key thing about this RXR motif is that it didn't have to be at the CMOS terminal. It actually could be nested within a loop. And so our first sort of goal is, okay, if we took our pre-existing release design and simply swapped out the KKMP, this dilysine motif, with the RXR, does this still function? And indeed, you know, we still get really nice control over protein secretion. And this really helps us unlock a lot of the potential of release in these polycistronic designs. So our next step was to actually implement the single transcript design. And I'm just showing you here, you know, we did a single transcript design where we had a split protease that could be activated in response to rapalog, which just is simply a heterodimerizing drug. We then, you know, attached a luciferase to the release construct, and then had a reporter protein downstream of it as well. And we just stably integrated into APRE19 cells. And the idea here is, you know, you would have without the rapalog being present, your protein of interest would be retained. When you have rapalog present, it would be activated. And what we ended up doing is we took these cells, we microencapsulated them, and then we just subcutaneously delivered them into a mouse model, a hairless mouse model. And the idea here is that we wanted to just simply show that if you gave a systemic drug, so in this case, rapalog, which is normally inert, could we control the local concentration of a protein of interest, in this case, luciferase? And our output was simply using IVIS to image when we'd have this burst of luciferase. And what I'm showing here is that, you know, after the injection, when we took readings every 24 hours, we saw that we could indeed get control of a protein of interest at the local level by using a systemic injection. And so this was great because, you know, you can imagine that if you wanted to be able to control the local concentration of other proteins of interest, you can do this in a really simple way. But now, you know, again, I always think, you know, how can we make our circuits more compact and simplify them even further? And one thing that, you know, we've really thought about is, okay, well, I talked about this situation where you had, you know, two signals that were pro-inflammatory and you wanted to respond to them in either an or and logic. Well, what happens if you have the situation where you have you want conditional protein activation or secretion? So in this case, you want, you know, TGF beta to be secreted when IL-1 beta is present, but the second IL-4 is present, you want it to turn off, right? So this, you can imagine this as a conditional protein secretion, and fundamentally this requires both a positive activation arm and a negative repression arm. And what I'm showing here is that if we wanted to actually implement this using, you know, the current setup of protein circuits, you would actually require two additional proteases to process this information and implement it. So the IL-1 beta is the activation arm. You have activation of the release construct secretion. The other case, you would have to use another intermediate protease to inactivate that other protease that would normally activate release. And so, again, in theory, this can work, but you can imagine that it starts to become quite cumbersome in the sense that now we require two additional proteases, which is, you know, kind of just starts to take up more and more space. And when we think about, you know, how do we actually begin to deliver these single transcript designs, you start to really realize that you're becoming limited by the size of your vector. Or even if we wanted to deliver this via mRNA, I think the current gold standard is around like 3.5 to 5 kb base pairs for mRNA delivery. You might be able to get a bit higher, but your efficiency really starts to drop. And so we realize, though, that, you know, what's lacking is this negative regulation of release. And so I'm just showing here that what we did is we tried to implement the CHOMP system and to have this negative release. So we have a HCVP inducible release, and this HCVP protease can be inactivated by the presence of TEV. And what I'm showing here is that this was supposed to be the NOT logic, and even though we're titrating, in a dish, we can carefully titrate the amount of TEV. We don't really get a full turning off of our system. So we realized that this really requires multiple proteases. The components need to be carefully tuned. We're increasing the size of the transcript, and also if we could simply reduce the number of moving parts, we're just going to simplify the entire system. So what our goal now was, could we modify release to achieve this negative regulation of protein secretion all at the release level without requiring CHOMP? And going back to the biology, the way that these retention motifs work is that they're recognized by the COP1 complex in the early Golgi apparatus, and it's simply this recognition that drives this this retrograde transport back to the ER. There are actually some proteins that can control their surface expression by phosphorylation and recruitment of these native scaffolding proteins called 1433 proteins, right? So the way it works is that when this protein is phosphorylated, you end up getting recruitment of the scaffolding protein, which is quite bulky, and it simply masks this retention motif from being recognized by the COP1 complex. And so our first goal was, what if we simply used an agonist against these 1433 proteins? There's an agonist known as R18 peptide. And so we did, with and without mutated R18 peptide to show, can we actually block this retention machinery of the RXR motif? And indeed, when you use the mutant version, so you have this you have COP1 still being able to interact with the retention motif, you end up getting retention. But when you have the functional R18 peptide and recruitment of the 1433 protein, you end up getting blocking of this retention capabilities, and the protein of interest is secreted. And so you can imagine now, using the same sort of logic, well, what if we simply put a cut site between the retention motif and the R18 peptide? The idea being here is that, when you have this R18 peptide present, 1433 proteins would be recruited, you'd have blocking of the retention motif, the protein of interest would be constantly secreted. And then when you have activation of your circuit or your protease, you end up removing the R18 peptide and then subsequent restoration of this retention capability. And indeed, we end up getting NOT logic, all at the release level, with really requiring a minimal addition of this R18 peptide. And so, we wouldn't be a SynBio lab if we didn't now go and create all the gates, which is fine. But what I want to sort of highlight is this NIMPLI design. And the NIMPLI design is really nice because it actually sort of fits this conditional protein secretion. And so I'm just going to quickly sort of go through the design where you can imagine this is the NIMPLI design, where when you have no proteases present, your protein of interest is being retained because of the N-terminal retention motif. This C-terminal motif is being blocked by R18, but we know that we can you need to cut both of those retention motifs in order to function. When you have the IL-1 beta sensor activated, you'll have removal of this N-terminal motif, and you'll end up getting protein secretion because this second motif is being blocked by the R18 recruitment of 1433 proteins. When you have the other protein being like the IL-4 being expressed, you end up just removing the R18 peptide, so the protein of interest is still retained. And finally, when you have both present, even though you cut that initial N-terminal motif, you also remove the R18 peptide, restoring the retention capabilities of that C-term. And so in this case, you end up getting this NIMPLI activation and conditional secretion of proteins. So the other thing that CHOMP does that I really wanted to get away from using CHOMP as much as possible is also this idea of quantitative activation for controlling protein secretion. So what this means is, can we use release so that we only have protein secretion within a very specific range of an input. You can imagine this being really useful if we wanted to have like subsequent activation of different proteins, right? You can have one protein activated, in this bandpass region, and then a standard release down the line. So this is a really powerful tool. And one thing we realized, and what's really nice about proteases, is that you can actually tune the catalytic efficiency of these proteases by simply swapping a bit of the protease cut site. So you can imagine that if you had something that had a high Kcat, it would take very little protease to turn off, in this case, the not Kate. But if you lowered the catalytic efficiency of the cut site, you can begin to shift at what concentration of input do you begin to have this cleavage event occurring. And so what we're showing here is that you can, this is quite easy to tune. We're simply tuning the negative arm, shifting the amount of protease required by increasing it. And so going back to our favorite NIMPLI gate, if we simply change the proteases so that they're the same, respond to the same protease cut site, so that they respond to the same protease, but tune their catalytic efficiencies so that the first cut site is more efficiently cut than the second, we can begin to look at developing this sort of bandpass activation. So in this case, when there's no protease or it's outside of that range, the protein of interest would be retained. Once you're in this bandpass region, only the first cut site would be cleaved because it's more catalytically efficiently cleaved. And this would cause the protein to be secreted. And then at high levels of your input, both would end up being cleaved, and then you have retention of the protein of interest. And indeed, when we design this sort of bandpass secretion approach, we can end up getting this bandpass activation of protein secretion by titrating the amount of protease. And so, this newer platform we're calling compact release, and I feel bad in a way because we're trying to get rid of using CHOMP and simplifying the system as much as possible. But you can also imagine that, by having the capabilities of programming at the release level, you can also begin to layer and have more complex circuit designs by, using logic at the CHOMP level as well. And so with that, I'd like to thank, the Gao Lab, Xiaozhen Gao. We started peak pandemic, April 2020. It's been quite a ride. Our collaborators in the Bintu Lab, the Apple Lab, as well as the Elowitz Lab, and my funding sources. And happy to take any questions anyone might have. Thank you.
Wonderful talk. Let me ask Mikhail whether he has comment or question.
Sure. Yeah. Beautiful work. Really enjoyed hearing that. Just very elegant design. A couple questions. One is, so what happens if you don't have the proteases or they're not active? Like, do you get just kind of continued buildup of the protein in the secretion apparatus? And that's the question. And then, how do the cells like that?
Yeah, so that's a really good point. So we've looked at the sort of cell state without proteases, and we find that the one really nice advantage of the release platform is that you don't really require very much to be delivered. So when we do our transient transfections, like we can get away with like 1 to 2 nanograms of a plasmid. And what happens is that we imagine, because we're using the native machinery, there is sort of like the endoplasmic, the ERAD pathway, which sort of creates a degradation of the protein of interest if there's an accumulation. So we have not seen sort of like a cell stress event occurring by not having our proteins being released. And when we've tried to really pump as much plasmid as possible, the cells seem to still survive, but what happens is that the retention machinery can't keep up. So you get sort of a leakier system. So I think because we're using the native machinery, there is some sort of safeguarding there that the cells can do. Yeah.
That's cool. That's very cool. Another question I had is I think the idea of responding to, let's say, pro-inflammatory cytokines and then releasing anti-inflammatory in the context of transplantation is amazing. And kind of in some of your slides, you had the inputs, IL-1 beta, IL-4, leading to protease activity. Is that something that already exists, or those are components that still need to be developed?
So some of those components are being actively developed. I think so. The Josh Leonard's group at MSBW had presented on actually, I think there was like a TGF beta sensor and IL-10 sensor, but we imagine that I've focused more on the processing and output modules, and I've kind of stayed away from the sensor design. But yeah, the short answer is we need to still develop these sensors. Whether or not we do sort of like a MESA design, that might be one way. Another thing, Xiaozhang's group has really focused on these RNA-based sensors. There might be some sort of interplay there where you can imagine that if you have an activation of a specific pathway or upregulation of a gene of interest, that might be one way to sort of repurpose the radar system as the input module for these protein circuits. I think the goal though would be to use like a MESA-like design. But no, I haven't actively tried to do that. I'm hoping Josh's group, which have so much more experience with that, they, if we went to them and asked, they might be kind enough to help us out there.
Are those working through transcription, or
so
can you see those directly connect to protease?
Yeah, so I think they end up they have a split protease design in some of their MESA designs. Okay. So I can imagine that we can sort of use that intermediate protease setup we did with the RAS circuit by simply swapping out the input being this MESA design split protease. There might also be a way in which we can use protease activity to recruit something to maybe block retention using maybe a 1433 like thing. That's still a little bit up in the air, but that might be a little bit simpler to translate. You can imagine simply swapping out the transcription factor with maybe a coiled coil, one of Jarala's coiled motifs, and sort of have that be recruited with maybe an R18 peptide. That might be one really simple way to implement that.
Got it. Am I allowed to ask more questions?
Yeah. Please.
Okay. Okay. Just one more. I also really liked, you had this picture in the beginning of the beta cells with filled up release granules just ready to fuse and release. Is there any hope for having something like that be the output of a SynBio controlled pathway?
Yeah. So I think that from the current release can do sort of, we can control insulin, but the tricky thing and the things that we haven't been able to really focus on is how can we make it faster? And I think the really nice thing that beta cells do is that their pre-packaging is a little bit different in the sense that they have these secretory vesicles ready to go. Mm-hmm. And the longest time I was trying to figure out how could we implement this in a way that you only package the protein of interest, because insulin's a little bit unique in that it the exact mechanism of how it forms these secretory vesicles is still a little bit up for debate, but one hypothesis is that it sort of via supramolecular interactions it sort of aggregates, and then in response to a stimulus it allows it to be secreted. So I'm still wrapping my head on how we can really create a platform in which we get those prepackaged secretory vesicles without affecting or controlling the secretion of other proteins of interest, like the native proteins of interest in the secretory pathway. So if you have any ideas, happy to chat, but I think that's like that would be a really nice goal, because I think we could speed up the response rate. Like right now we can start to see differences in secretion at about 3 hours after activation, which sort of coincides with protein secretion in general from the ER. But we always are looking to to find ways to make it faster.
Okay, great. And then finally, I'm just curious how much debate you and Xiaojing had about whether to call the negatively regulated system release not versus coming up with an acronym for retain.
Retain. Ooh, well, we haven't published this yet. So, and we're in the process of pre-printing it, but retain might, you may be on to something there, Mikhail. I might have to put you as in the acknowledgements for the acronym. We got to figure it out, but
don't worry about the acknowledgement. I'll have internal satisfaction.
Yeah, no, retain. Actually, that's yeah, we just kind of went with release or release me not or whatever, but retain could work. Yeah, I like it.
All right, nice, great talk. Really, really beautiful work. Thank you.
Yeah, thank you so much.
Thank you. I didn't see any question from audience, so let me ask some question that actually I asked that question to me myself. So I used to believe in, big complex circuit is better. And then you also mentioned something similar earlier, like, simple is better. I want to kind of ask your perspective or insight based on your experience so far. What do you think? I mean, which one is better? I mean, the simpler circuit or complex one? Of course, that depends on what kind of application you're talking about. What is your view now? I mean, after many years of.
Yeah, I think so a complex circuit might be really useful in sort of interrogating certain pathways or behaviors. But I think from a therapeutic standpoint and actually trying to translate these things, I think I really fundamentally think that the simpler the system is, the less chance that something can go wrong, right? So I can imagine that with some of these really complex circuits, if one component for whatever reason, gets silenced or turned off, that can really mess up the entire behavior of the cell.
Right.
And I think one thing that I've really focused on is really simplifying it as much as possible with the goal of making it as translatable as possible.
Right.
So one thing that the release platform, because it really harnesses native and machinery, we've been able to put this into different cell types, and any cell type we put it in so far works. So I can imagine some of these more complex circuits or complex designs, you have to do some sort of optimization.
Yes,
I think so. I think it really simpler, the simpler the system, I think the more robust. Right. But you do definitely lack some of the programmable capabilities or really hardcore computational capabilities that a more complex circuit might be able to give you.
Yeah, I absolutely agree with you. I mean, the reason I'm asked that question is I used to think the other way around, like complex is better. Yeah. But now I believe for the practical application, actually that we need to deliver now, because after 23 years of synthetic biology, I feel that way. That synthetic biology start with the Jim Collins toggle switch and Lloyd's oscillating circuit, and another one is a negative feedback loop, all published in Nature. And then now 23 years, we should deliver something real practical, product that could be therapeutic, whatever.
I think though, like, so again, it kind of depends on how you look at it, in the sense that, like, so from a tissue engineering standpoint, there's no reason why you cannot deliver, 3 different engineered cells that have each a simple circuit inside, but they each have different functions. And so, then you have to think of, well, how do they communicate with each other? And sure, that's a different engineering capability altogether. But, I don't think you necessarily always have to think of having a single cell that can do everything.
Right.
Because that might be sort of limiting its translation. Like if you have a really robust platform and you have 3 different, 3 different outputs that you want to have, and you have 3 different cells, that might be okay.
Yeah. Yeah, so now I believe simpler the better. The simpler the better.
Yeah,
I have the
yeah.
one question from audience, Maya. She said, I may have misunderstood, but instead of reconstituting a protease on sensor dimerization, can you use MESA or a SynBYSS system to release the protease on activation?
Yeah, so I think that could be feasible. The only tricky thing with sort of having a fully functional protease, being present on, let's say, a receptor, or like a SynBYSS design, is that since they also will go through the secretory pathway, there is a chance that you'll increase your background because you'll have cleavage of the release construct as the protein's being translated. But there are ways you can sort of look at caging proteases, so that you might be able to mitigate some of that potentially, increase in background. So I think you can either use a relay protease or find some sort of way to drive this localization of a protease to the ER upon activation of the receptor. Yeah, I think it's doable. I just, I don't know the best way to do it, but I think it's feasible.
Okay, let me double check whether we have another question. I don't see any. Okay, so it's 11 o'clock my time. Let's close.
Okay.
So thank you all for joining and staying today. We'll meet again next week on August 17th, Thursday, the same time, the same Zoom link. We'll have Professor Ron Weiss at MIT and Dr. Jennifer Nash at MIT as well. As usual, the following informal chat will occur without recording. Please stay here if you are interested in chatting with us. We'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I will stop record. Okay, just give me 1 second.